Hello everyone, and welcome to the first program of a series on Somatidian Orthobiology. To speak to us on the subject, we have invited Mrs. Jacinthe Levesque, biologist. She is also the director of the International Academy of Somatidian Orthobiology. Mrs. Levesque, it's a real pleasure to welcome you to the program.
Thanks for this invitation. We're very happy to be part of the program.
Mrs. Levesque, I have had the occasion of meeting you several times before this program, and I have noticed your great interest for Somatidian orthobiology. How did you get personally interested on the subject?
Well, I guess we cannot hide this interest. It's probably very obvious. From my point of view, I think that my arrival to the Somatidian orthobiology is probably a very logic evolution of my career path. Actually, I'm a biologist. This is my basic training. So, of course, being a biologist, and anything that has to do with biology is of great interest. So this is probably my prime training.
Did the interest start at a very young age?
Well, I would say that the interest, like many other things, started probably 10 years back. So it's something that, progressively started to grow. Also, I have a training in pedagogy, which considers all the, I would say, the learning process and all the diffusion and all the techniques. So I guess also this interested me because the Somatidian orthobiology at this point is in diffusion everywhere in the world, and I think we need all these tools to help to bring out the theory. Also, I have a training. I have a master degree in public health, which is very much aware and interested in all the prevention of disease and how disease is explained with various population patterns. So I guess all this put together, it seems for me that the Somatidian orthobiology is probably something that creates a bridge between all the various fields of interest that I had in the past. So it seems a very logic evolution from my point of view. Now, of course, I've been a director of the International Academy for some years, and well, one thing leading to another, and because of various circumstances, it led you. It seems to have a very natural progression. And I think, as you've mentioned, I'm very happy, and it's a very...
How long have you been a director of the Academy?
I've been at the head of the Academy now since its opening. Maybe it's 3 years now.
Of our viewers, and also to give us a better idea of what we'll be discussing within the next couple of weeks, would you give us a brief description of Somatidian orthobiology?
I think it's a very good idea at this time. We will probably see it more in detail, but for now I think we could very well say that the Somatidian orthobiology is a new biological theory which studies life phenomenon in regards with the evolution, that is, with the changes of form of a new particle discovered in 1954 by the French biologist Gaston Naessens. He later called this particle somatide. So if we take the expression orthobiology and we just see from the Greek origin, the Greek root of this word, we see that ortho means to correct, to straighten up the biology in consideration with the knowledge of the somatid. So Somatidian orthobiology is a new theory, is something new in the field of biology.
How did the scientific community react to this discovery?
Well, we can say that this discovery was greeted like any new discovery, I think, that happens either in biology or in the scientific fields. Actually, as you know, science is a very conservative environment, and I think it was very normal for the scientific community to just let time go by and see what would happen, and this is a very normal procedure in science. I mean, once you make a discovery, I mean, it's exciting for the one doing it, but I think it has to be...
There's a lot of questions.
A lot of questions. It has to be looked. There's a matter of time. It has to be repeated by other researchers. And I think in general we can say that this new discovery was just greeted like any new discovery that happens in the biological field. Now, even for Mr. Naessens, who himself made the discovery, I think it's important also to understand that at first, of course, he was a little bit stunned to see all this, but he needed time to look at all this to make sure it was not contamination, to make sure that the particle was not an artifact, either created by a light or created by the preparation of the samples. He had to check if the particle that he was seeing was not a lipoprotein or chylomicrons. And finally, himself, as the researcher who had done the discovery, he had to be very much aware of all the environment that was happening, and he had to take time and make sure that before, bouncing up on the walls and being very excited. And in general, we have to be very cautious, and this is, I think, part of the philosophy of science, that even though something seems exciting, you need to take time to look at it very closely and to make a very close study of the particle and all the new information that you're bringing up into the scientific field.
Now, we speak about Mr. Naessens, and I think at this time it might be interesting if you could speak to us about the highlights of his career and also maybe a little bit about his personal life.
Well, I think it can be interesting just to follow how this researcher, actually, as we said earlier, Mr. Naessens was born and raised in France. Actually, he was born in a city called Roubaix, which is in the northern part of France. He was born in 1924 in a very typical French family, actually.
Did he have any siblings?
Yes, he did, because he was the baby child of a family of 4. And it's a matter of fact, his older brother, right before him, was 9 years older.
Oh, really?
So Mr. Naessens was on a way in a big family. There was 4 children, but he was sort of brought up like a unique child because all his brothers and sisters were much older than he was. So this is probably what gave the character of Mr. Naessens of being a very solitary. He's independent and he's very autonomous. I mean, he was sort of brought up to do his own things and play by himself, and it probably gave him the character that we recognize of Mr. Naessens. Also important to say that very early, what we can say, he was 11 when his father died. His father was a well-known banker of Roubaix, so very early in life he lost his father. And maybe another major event that had a lot of influence on Mr. Naessens' character.
It's a very important age, at a time to lose your father, your dad.
It's like anything in life. We are always being influenced by something. And in its case, war, the outbreak of war happened when he was also a teenager. So this also is probably a very important event that has created an impact on the life of Mr. Naessens. Actually, when he was in 1939, the family was living in northern part of France, and in 1939, like many other French families, they have to evacuate. They had to evacuate southern France. So Mrs. Naessens, her children, including Mr. Naessens, they had to leave their home and departed with almost nothing, and they left, and they which is not easy, and they moved southern France, and they got organized and started a new life. Now, at that time, Mr. Naessens was 15 years old. He was a teenager studying at the University of Lille in a scientific program, and because of war, well, everything stopped for a little while. His training stopped. And thanks to the initiative of the same group of teachers that were teaching at the University of Lille, who also had to flee down south because everybody moved down, they started a new institution, like a new temporary educational institution. And because of this initiative, he could continue his training. So I think this was a very positive thing that happened. So that in 1945, actually, he finished his training program at the Union Scientifique Nationale Française, which was a very well-known
respected and well-known
at the
institution.
time, but considered a temporary educational institution.
So not recognized?
Well, it was recognized, but it was something that was being done just because of war. It was like a very temporary situation. And he was graduated then in 1945. He received his engineer degree in biology. And let's say that's how he got his training. Now, because of war, because of all this situation in France, he got all these things organized, and finally in 1949 he had his own private lab, and that's where he started to work on his own.
That was still in Europe?
It was still in Europe at that time, and he continued to do his research. And even though as a student he was always attracted towards the blood. So the blood was always, from the beginning, something that he would very, very interested and concerned. So as a new, I would say, graduate in biology, he started his hematology lab, and also having a little, I would say, hobby at that time. He, because of all his aptitudes to work, his hobby was
He was mechanically inclined, I think you told
me. He was very young. Maybe we can add that very young in life. I think he was 4 years old. His mother surprised him trying to powering his mechanoworks, you know, with the various tools of a family alarm clock. So I think he was quite a challenge for his mother. But you know, that was probably a very positive aspect, because it gave him all what he needed, and as an adult, actually, he decided he would like to work into the microscopy field. So this was sort of a hobby, and one thing leading to another, finally the hobby became almost a passion. And this is also probably a very important turning point in Mr. Naessens' career. In 1951, just like this, he decided to upgrade the performance of the optical microscopy at that time, all the microscopes. So he gave himself a challenge, just for himself, just to see where it would lead. And finally, he developed a very unique tool, which he later called the somatoscope, which gave him a high performance and was very practical because of his work in blood. It made him see blood through a completely new vision. Now the somatoscope is a tool that he has been using since then to do his research. But at the time it was just like it happened, and it was very practical. Now this particle, as we have said earlier, was discovered in 1954. So since from that time on, starting from 1954, he saw it in blood, but now he had to isolate the particle in order to put it in culture and see what was all about this new particle. Now from 1954 to 1959, which is a lot of years, this is the time he took to be able to find a way to isolate this particle and put it in culture, which we will see eventually later on in the program. Now this brought him to 1959, and from 1959 to 1963, that's the time he took to just look at the particle, understand it.
4 years.
4 years to understand the understanding of how it would behave, what was all about, trying to put different hypotheses and trying to find actually what was the function of this new particle in blood. You know, nature is so well done that if this particle was there, and as he could see it very clearly, there had to be a reason. It was not there randomly.
Right.
So
It didn't happen.
It didn't happen. So he was really concerned and had to find what was the function of this particle in blood. Actually, his theory was submitted to the Académie des Sciences de Paris in 1963. So this is a little bit, I would say, a resume of the history of Mr. Naessens' career.
It's really very interesting. Very, very interesting. Now, you mentioned the somatoscope. Now, what is the greatest quality of this instrument?
Yeah, I think it's very interesting. Before I walk into Mr. Naessens', I would say, marvelous tool, which is the somatoscope, it's maybe important to understand that when you talk about any microscopic tool, Okay, there's two qualities that we're looking for. We have to want to see that a microscope is a support. It's a tool that we use to help the human eye to see better. So one of the major qualities of a microscopic tool that we can expect is what we call the power of magnification. Okay, I think it explains how big is magnified. How big can we go and see this tiny particle that's usually invisible to our naked eye, but once we bring it on the microscope, it becomes clear, and then we can see what we're looking at. This is one major quality. The other quality that we're looking at when we talk about microscopy is what we call the power of resolution, which in the field of physics means how clear is the image. How clear, because I mean you can magnify as much as you want, but if you don't have a clear image, you're going to have a blur, and it's not going to give you that much more information. So if
that equipment
is very, and this second quality is also very important. And in fact, Mr. Naessens' somatoscope, the tool that he has designed and done himself, is very performant in terms of resolution. At the time, the optic microscopes that were available at that time in the 1950s usually gave a magnification power of 2000x. Now, Mr. Naessens' somatoscope could bring the magnification power to 30,000x. So this was a major improvement in terms of magnification. But not only this, in terms of resolution, the highest that we could get at that time was 2,000 angstroms, which is the unit with which we measure the resolution. Now, his somatoscope brought the resolution power to 2,000 to 150 angstroms. So this was an improvement of 13 times, 13 folds better in terms of clarity. So this was a major improvement. So all this combined permitted to see better, to see with so much finer details, and above all this, I think another quality of Mr. Naessens' somatoscopes, that his tool permitted to see live material, which was so important at that time. We have to understand that we're in the 1950s here, and that was like the arrival of the electronic microscopy, which was the new a major breakthrough in terms of microscopy at that time. But the only thing that the electronic microscopy requires is that we dehydrate the specimen. And once we dehydrate the specimen, we lose the capacity to see the somatids. So at the same time, in the same period, Mr. Naessens upgraded the optic microscope with his tool, and on the other hand, it was the arrival, it was the new thing of that period, the arrival. Now, of course, the electronic microscopy is much more performant than the somatoscope, but it does not permit to see the live specimen. So if we summarize all this, we can say that Mr. Naessens' somatoscope is probably a tool halfway between the old model of the optic microscope and the new arrival of the electronic microscopy.
Will we be able to see this during this program, how it works, or?
Most likely, and I think maybe for the purpose of the for the viewers, maybe we could just have a look at the underlying principle of this tool that we have called the somatoscope. Maybe we can just look at the screen.
Wonderful.
The underlying principle of this apparatus is that the frequency of the observing light can be greatly increased. Two light sources, one incandescent with a wavelength of 3,300 angstroms and the other ultraviolet and set at 1,850 angstroms, are mixed together to produce a third beat wavelength. This resulting wavelength is treated by a monochromatic filter to produce a ray. The ray is subjected to a magnetic field and split into parallel rays, the Zeeman effect. One of these parallel rays is run through a Care cell to boost its frequency. This light ray, invisible to the naked eye, analyzes the sample to be studied. Using the somatoscope, we have been able to observe within the blood an elementary particle endowed with an electronegative movement of repulsion. We have called this particle the somatid. The particle is minuscule to the extreme, varying in dimension from a few angstroms to about 1,000. Still, with the somatoscope, we have been able to observe its polymorphism clearly.
That was the most interesting information that you just showed us. Thank you so much.
Well, it's quite revealing. Maybe at this time we could probably use the occasion to show, now that we have seen the somatoscope,
Right.
maybe we could look at a few live samples just to give a little.
Oh, you have that?
Yeah, we do. A little preview here for the eyes of the viewers,
Okay.
because it's quite a good image to look at. Here we have a sample of fresh carrot juice, just to show you the variety of colors that we find in the different natural samples.
This
Here
is
we-
carrot juice.
Yeah, carrot juice, just taken freshly from the carrot,
All right.
put between slide and cover slip, and we'll look at it on the somatoscope. Now, the colors that we see here are natural colors. These are the pigmentations found in the carrot juice, chlorophylls and all these. Now, here, this new sample that is just coming up is the hemolymph of a dragonfly, which is another type of live sample, just to show you the variety of samples that we find in the nature. Colors are very clear, and
Forms are a bit different.
forms are a little bit different, but the idea here is just to show you that in every sample that we're looking here, in the live form, we are always seeing the little dancing particle in the background.
Right.
The same movement is there, and it's always present, because we have found out later on that the somatid is a particle that is present in all form of living matter. We have seen the carrot juice. Now, of course, this is a vegetal, okay? It comes from the another kingdom, the dragonflies from the animal kingdom.
Right.
And now on the screen we have here the picture of live blood, actually. Now, for now, I think it's only important to check the little dancing particle. It's the thing that we want to show at this point.
The tiny, tiny
The
part.
tiny particle that you see that's dancing around, and it's just have a very special movement. Of course, here we see the blood cells, but in another program we will have the chance to explain more in detail how does the blood look.
I see.
So what's important to retain from this experience is that the somatid that we have seen in the vegetal kingdom, the one in the dragonfly, the one in the blood, is always presenting the same little dancing movement and is always present, although we are talking about different kingdoms.
Now, what circumstances led to the Somatidian theory?
I think we can summarize all this by saying that Mr. Naessens, because of his interest towards hematology, decided to work more precisely with that tissue, and because of his personal aptitude to work in mechanics, and because of his hobby that he had to create a new tool, finally it led to the discovery of a particle. Now, with that knowledge being available from that time on, he decided that he would look forward to see exactly what was this particle. So I think we can summarize by saying that a man had an idea, he created a tool, discovered a new particle, and by observing this new particle, wrote a theory, and that's probably the summary of the evolution.
So that's what led to the theory.
All this put all together led to the theory.
Wonderful. Unfortunately, Mrs. Levesque, our time is running short. What we'll be talking about next week?
Next week, we will see how Mr. Naessens has isolated this particle, and mostly what revelations did he get from this experience.
Well, I really look forward to this program next week, and I thank you for being here on the show today.
Thanks.
Hello, everyone, and welcome to the second program of a series on Somatidian Orthobiology. To guide us on this fascinating voyage in the world of Somatidian Orthobiology, we have with us Mrs. Jacinthe Levesque, biologist. She is also the director of the International Academy of Somatidian Orthobiology. Once again, it's wonderful to have you with us. And Mrs. Levesque, where are we on this extraordinary voyage?
Well, it's a matter of fact, we have last week seen a lot of things, and I think for the purpose of the viewers, we could probably do a little short summary here. I will recall the definition of the Somatidian Orthobiology, which in fact is a new biological theory studying life phenomenon in regards with the evolution, that is the changes in form of a new particle that has been discovered in 1954 by the French biologist Mr. Gaston Naessens. Later on, he has called this particle the somatide, and this is a little bit what we have seen last week, that with the help of the somatoscope, which is a microscope that he has designed and built himself, he has brought up to light in the blood, actually, what he has called the somatide. Now, for the purpose of the viewer, maybe we should go back and just give a slight image of how, for the eyes, how does it look when you look at a conventional blood smear compared to the picture that we can see when we look at live blood. I think it's already on the screen right now. Now here we have a very conventional picture of normal blood. You can see the red blood cells and the white blood cells. Now on this right hand, a half of the picture, now we can see the image of a live blood. Now we will see later on, I will comment a wider picture a little bit later on, but the important thing here is to understand that the somatide is this little dancing particle, this little mobile, very motile particle that we see in the background that flows in the plasma and drives along between the blood cells.
Right.
Now, of course, blood is a very interesting and fascinating tissue because, as we know, blood is the only liquid tissue that we have in the body. And very fortunately also, we've said last week that Mr. Naessens' microscope had the possibility to look at live material. So, of course, blood is a very good tissue to look at. And eventually, we can say that blood is a little bit like a highway in the body. Anything that travels in the blood has its own purpose. It's either a nutrient or something that is going somewhere to feed the cells, or it's something returning from the cells, either a waste product of whatever kind. So everything that's traveling in the blood has its own purpose. Now I think here we can probably look here back on the screen and we will see blood that has, it's a sample of fresh blood here. And we will probably comment on blood cells because, of course, we have the somatids, but the nice...
Tiny particles.
Yeah, the tiny little dancing particles that we have just identified previously. But here, all these nice clear circles here that we see, very clear, very round, are what we call the erythrocytes. These particles are commonly called red blood cells.
There seems to be a lot of them.
There is a lot of them because this is probably the cell that we have in our body in a very much numerous quantity. Now, here we can see that the membrane of this red blood cell is very clear, very refringent, very, and this is because of its content in hemoglobin. We know that the red blood cell has one purpose, mostly to deal with gas exchange, either to pick up the oxygen from the lungs, bring it back to the cells, and to return the metabolic waste product, which is the carbonic acid, and bring it back to the lungs. So this type of cell is very much in link with all the gas exchanges in the body. Actually, here in the center position, we can very well see a white blood cell. Actually, it's a neutrophil from the family of leukocyte, which we call commonly the white blood cells. Now, this type of cell, this one purposely, is very much involved into the bacterial infection. So this one is just like sneaking around, sniffing to see if there's any type of foreign body in the bacteria family that he could just sense, and if he sense, This particle is just being grasped and being digested.
Takes action.
So it's very interesting to see here that this white blood cell, which is usually called our soldiers, is very active. So this is an example to show that when we look at live blood, we can very well see the interaction, the movement of the different particles, which is something that we don't see with conventional blood tests. So it's another aspect of the same tissue that we're looking.
But you have different types of white blood cells.
Yes,
right?
actually, you're right. We have various types. This one is one kind, but there's actually 5 different kinds of white blood cells. All of them are working as soldiers to defend ourselves from foreign invasion, but every type has a specific function.
Oh, I see.
We might see in our sample, if we're lucky, different samples here, but we have to go with, we see here. Neutrophils that we see here are usually the highest population of white blood cells. Usually they represent 60 to 70% of the population of our white blood cells that we have in our body. You see here the nice movement that we see. The white blood cells sorts of throw away the parachute, brings all the granulations, and we see it just moving around, and this is life. It's amazing every time the activity.
Fascinating. It really is. I mean, to see it on the screen like this.
Yeah. And here at the 3 o'clock position, I don't know if it's clear, but it is for us, but for the viewers, we have here platelets, which is another type of blood cells, mostly involved into the coagulation process. This type of blood cell is very important in the event if we cut ourselves or if we broken up any type of our capillaries or vessels. It just creates a clot and prevents the blood from overflowing and prevents dehydration that could happen. I think we have a platelet here just side by side with the neutrophil. They're very small. The platelets are way smaller.
Yeah. They're not as easy to
to recognize. Yeah, usually it's. So I think this is a pretty good picture. And here again, let's recall that the little dancing particle here in the background, the little, very brilliant spot that's moving around. This is the famous particle that was discovered by Mr. Naessens and that he has called the somatide.
If any of our viewers would like to pursue their learning experience on Somatidian Orthobiology, or if you wish to have any additional information on the topic that were discussed on the program today, I invite you to contact directly the International Academy of Somatidian Orthobiology by calling the number that appears at the bottom of your screen. That was most interesting information you just gave us as we looked at the screen. But from there, how did Mr. Naessens pursue his research?
Well, actually, what we have just seen is the blood picture that Mr. Naessens saw in 1954. We can say that it was pretty much the same picture that was revealed to him at the time he made the discovery. Now, of course, it's very interesting to see a new particle, but from there, he really had to find a way to isolate this particle, because this is happening in the blood. In order to better understand how does this particle evolve by itself, what is the behavior of this particle, you really needed to isolate it from blood and from there try to put it into an environment which would permit this observation. Of course, this particle is being so small, we know that centrifugation, he knew how to sort of isolate other blood cells, but this one was sort like he was doing pioneer work at this point.
So how did he isolate the particle?
Well, that's the
key question.
The key question here. Actually, because of this new discovery, he had very little place that he could look. There was really no recipe he could follow. Nobody else had done it. So this part of his research actually took a lot of years. Actually, it lasted from 1954 till 1959. All these years, he tried actually to isolate the particle. At first, he did a little bit like we usually do in conventional blood analysis. He used a centrifugation, which is a very fast spinning of a sample, and usually because of the weight of the particle, you know, it just separates, the different types of blood cells separates. And at first he thought that would be the answer, a little bit like we usually do in different blood cells. But he found out that this was not working like he would, and
wasn't happy
it wasn't.
with that.
No, it wasn't really happy because he couldn't have a pure culture. Finally, he used after that he used another method of, you know, a little bit of foam, and he was hoping that because of the lightness of the particle, this particle would just, you know, jump into the top of the specimen. But again, this didn't work out. He used another technique. Actually, he burnt the whole specimen, hoping that all the blood cells would disappear, and finally he would only have the somatid left. It sort of worked out, but finally, from one thing to another, he finally adjusted a technique, a proper technique, which is part of his research work. And finally, he got this particle isolated from the blood, and now, well, ready to go on with his research. And now we're just about in 1959 at this point.
And what is that technique called that he developed?
Well, actually, there's really no name specific for his technique. I think this is part of his specific work of Mr. Naessens. But all we can say is that once this particle is isolated and extracted from blood, it's a pure sample of somatid. All there is somatids. And this was mostly needed for him to continue his research in respect with all the scientific equipment procedures that we need. In science, if we want to look at a particle, we need to isolate it and put it into what we call the culture environment.
A culture environment. Now, what is that, a culture environment?
It's a specific, I would say, expression that we use in the field of biochemistry, actually. A culture medium is a very simple environment into which we find nutrients, good nutrients,
Right.
for life to express itself. And it's a condition, it's an environment that we control in terms of temperature, in terms of pressure, in terms of whatever we want to check is being controlled very tightly. So basically a culture environment is a media, it's an environment. In this case we're talking about a liquid environment that Mr. Naessens used. It's a simple environment where there is food, there is nutrients, and of course we need to be sure that the environment is not contaminated. So it's a pure, sterile environment in which there is the nutrients needed for life to express itself.
So in other words, all of the elements needed for this medium culture have to be perfect.
They have to be perfect. It's like a nurturing. It's nurturing this life cell and helping it to grow, if it is to grow, because at first he wasn't sure that this particle was alive. So let's say it's the ideal condition into which life can express itself.
So once he has it in this perfect environment, this culture medium, what did he observe?
What did he see? Now this is where it gets very interesting. Matter of fact, Mr. Naessens, when he took the somatid, put it into in culture, and let it go, at first he had to let it go, 90 hours later, okay?
How many hours?
90 hours later, there was already revelation of something very interesting. And I think for the purpose of the viewers, we will put on the screen a diagram of what
Right.
Mr. Naessens has seen as an overall picture of what was happening in culture. Now, one interesting thing to see here in this diagram, and interesting and very important for viewers to understand, these what we're seeing right now are images. They're not true pictures of what we see in cultures, but this is only a tool for us to understand guidelines to see what's happening. Now this has been drawn after the experiment has been started thousands and thousands of times. because, you know, as any new discovery in science, you don't just start to draw anything. You draw things or you confirm the way it happens once you are sure. So it has needed repeated experience, and this is the overall picture that we have right here. The first thing I think that we can mention, we will start to see this diagram as a whole. Let's see it as a whole first, and then we'll go back to finer details.
All right.
One important thing here to say is that the somatid, you know, the pure culture of somatid, which was a very tiny particle, as we have said previously. We're talking here of a particle of very small that could measure between 150 to 1,000 angstrom, which is very small, at the limit of what the somatoscope can see actually. It starts to be a very simple particle, and progressively it goes into different forms, evolving progressively from a very simple particle into a very more complex figure. Here, interesting to see that the forms are always arriving in the same pattern. This was something that was very revealing to Mr. Naessens, that every time he started the experiment, the forms, as they come, always happened in the same pattern. So much that at the end we have a sort of a cycle. So this is why he has called this the Somatidian cycle, because it appears that the particle in the environment called culture creates an image of different forms evolving from one going to the other.
And I see from your diagram that some of them are, let's say, that they get longer and longer. Is there
You're right.
a reason for this? What happened?
Well, actually, we cannot explain the reason for it, but we can at least just see that it exists. That's the way, that's the facts, that's the way it appears. It happens very clearly that from a single form, very simple, it evolves into a very complex. Now, 2 times in the cycles, if you can just give a brief little look at the phase 11, which is the bursting of the mycobacterial form, we see here that there's a bursting that brings into the environment new little particles for the first time. Again, at phase 16, at the mycelium form, we see another phase that we call a bursting. So in the midst of this whole cycle, we see at 2 different occasions the occurrence of new little tiny particles, which are of the same nature as the one we have first seen, and are continuously doing the full-blown cycle.
Right. Now, what is this? Is this bacteria? Is this harmful to the health? What happens?
Very interesting, your question, because as we have said, different terms. Starting from this phase here, the first phase, Mr. Naessens has seen that these forms pretty much look like bacteria. They were shaped like bacteria. But the true nature of these particles, they're not bacteria. They're not they're not an outside structure that comes within the body. This structure grows within the body. So even though it is called bacterial form, it is important to recall that it is not a true bacteria. Same as the yeast-like form here that we have. It is not a true yeast, and same as the mycelium form. So it's the choice that Mr. Naessens took at that time was to name these forms according to the
identify their growth
or their form, because here we are just looking at forms, and according to the similarity they have with already known structures in biology, he called them bacterial-like form. Of course, if you look at it, it's true that it looks like a bacteria.
Yeah.
But if you look very closely, we have to recall that all this cycle here first started with a particle that comes from within. This little particle, the somatid, responsible for this whole cycle, comes from within the body. So it is not an outside bacteria.
Not an outside force. It's within us.
Within us. All this can occur within us. But remembering that we are still in culture here. We're just trying to see how this figure evolves. Now, one thing very important that Mr. Naessens has looked, we have talked about a culture, which is an ideal environment.
Right.
In the course of his experiment, he purposely changed the condition of the environment. He put some, let's say he put some chemical compounds that has brought this environment not ideal conditions, but let's say brought some very adverse conditions.
Right.
When the somatid is exposed to very hard adverse conditions, it has the property to retract itself and stay into a resistant form.
Really?
And this was something very important in the understanding of the somatid. Now later on, a few years in the midst of Mr. Naessens' research, he took this form, which is the resistant form called ascospore, and then he submitted again this form to very tough conditions in terms of temperature, in terms of chemical compounds. He even submitted this to radiations. So
and what happened?
the darn little particle could not even, couldn't kill. It just
stayed alive.
It stayed alive. So from this point of view, we can say that the somatid has a property of retracting itself and staying for long periods of time, and it's almost an eternal particle at this point.
How interesting. Now, what should we remember from his observations in the culture?
Well, I think the three basic things we can summarize this experiment by saying that the somatid is definitely a live particle. That's very important. It has the inner capacity to change its form depending with the environment. According to the environment, it changes its form. This phenomenon is called polymorphism, and we will have the chance to speak about this later on in upcoming programs. And of course, this particle can retract itself and become into a resistant form. Now, from all this, what's important to understand and to remember and grasp very clearly is that Mr. Naessens, by looking at this cycle, saw some figures, example this figure, ascus form, saw some figures here that he had seen for many years in the blood of sick people. So by looking at this, it really uplifted a very deep concern and a lot of questions in his mind. Maybe he was on a lead to make a link between these forms and the health status of one being.
I'd love to know what new information did the observation of the Somatidian culture reveal? Can you tell us that, please?
Well, I think that 3 points, 3 facts were revealed by this experiment. First, the somatid is very well a live particle. This was a major revelation, I think, to find a new live particle. Secondly, we could very well see that the somatid has this inner capacity to change its form depending on the nature of the environment. So this also was a new information, that the somatid had the what we call the capacity to express itself into polymorphism or pleomorphism. It's the same word. And at last, what was finally revealed is that the somatid has a capacity to withdraw itself and stay into a resistant form. So this brought a lot of concern for Mr. Naessens, because it certainly troubled his mind and made him think a lot about the origin of life on the planet, because all this new information just was so new and, shook up his idea so much that it was really a major revelation at that point.
Mrs. Levesque, what's on the agenda for next week?
Well, next week we will see from the information that was gathered with the experiment of the Somatidian culture, what can we expect now with the blood, with all these new questions that were raised in Mr. Naessens' mind concerning the presence of the form and the confirmation or not of the disease state. So I think we will explain how I would say the basic research that was done in the lab until now will now become a clinical research. We will need to go and check certain things on human beings to make sure that we understand well the presence and all the pertaining of the presence of the somatidium forms in the blood of someone. Now also what we will also see very briefly, but we will also present the different applications of Mr. Naessens' discovery, which is the somatid. How did he applied all this knowledge into the field of public health?
Thank you. It sounds like a fascinating program, and I definitely look forward to having you back with us next week. Hello everyone, and welcome to the third program of this series on Somatidian Orthobiology. To speak to us on this subject, we have Mrs. Jacinthe Levesque, biologist. She is also the director of the International Academy on Somatidian Orthobiology. Hello, Mrs. Levesque, and welcome to the program.
Thank you.
Now, for the benefit of the viewers who may be joining us for the first time today, would you give us a brief definition of what is Somatidian Orthobiology, and also perhaps speak to us about what we discussed on the first two programs?
Yeah, I think it might be very useful. We'll have a little review here.
Okay.
We can say that Somatidian Orthobiology is a new biological theory studying life phenomenon in regards with the evolution, that is the change in form of a new particle discovered in 1954 by the French biologist Gaston Naessens. Later on in his life, he called this particle somatide.
Mm-hmm.
In fact, in the first program, we have seen that throughout this, the career of Mr. Naessens, he designed a very unique special microscope,
Yes.
which has permitted the discovery of this particle. In fact, this tool, which he has called a somatoscope, is a highly performant optic microscope that can reach a resolution power of 150 angstrom, which is 13 times better than any actual optic microscope. So this tool has permitted the discovery of a new particle. Now, once he has seen this particle, which this is probably the content of our last program, we have seen that to go forward with scientific research, Mr. Naessens really needed to isolate this particle from the blood and place it into a very, I would say, culture environment to permit the growing, just to see what would be the behavior, what would be the new information that the growing of this particle would give us, would inform.
If I remember, it had to be a very specific environment.
Yes, we can just recall that in order to see if a particle is alive, we need to subtract all the environment except what is fully needed for this particle to grow.
Okay.
So it's a matter, it's very easy to understand. He took blood from a human, he did an extract, and with time we have seen also that this has taken many years to find a very specific way to do the extraction of somatids. And once he had find this out, it has permitted to see how it evolves into the culture environment. Mm-hmm. Now, what this experiment has gave us as for new information, we have seen that the particle, the somatid, was well alive. This was the first observation that this experiment could inform us. Secondly, we have seen that this particle had the capacity change its form depending on the environment. And
Mm-hmm.
that was like a new, it's brand new information, because it's a very unique process in biology that we call polymorphism. It's the capacity of a structure to change its form depending on the quality and nature of the environment. And thirdly, what was also revealed is that the somatid, if you place it into very adverse conditions, if you change the environment, if you place it in very difficult situation, it has the capacity to retract itself and stay into a resistant form, and this eternally, because once it's in the resistant form, it doesn't move. It's very hard. You cannot break it, you cannot burn it, you cannot, even we've seen last week that it was submitted to very strong radiations, and there's really nothing to change the property of this particle once it is in the resistant form. So this is pretty much what the experiment has brought as new information.
How does this change in form occur?
Well, actually, it might be easier for us maybe just to have a little glimpse here on the screen, and we will see a pleomorphic phenomenon arriving. Just have a quick look at the structure inside. You see at the middle position, this bacterial-like form, a little bit like a rod shaped. We can almost see the... you've seen it has just changed, and this is in real time. We just here have a glimpse of it. That's an example of a change in form, the pleomorphic process. Now here again, we will see that this particle will be capable again of changing its form within always according to the same membrane. It does not necessarily divide. So after all, we see that pleomorphic changes are always an extension of the membrane that is there. It's just a change in the structure, which with exactly what we have seen with the somatids. Now here we have exactly the same round structure, as you can see, has divided itself into three structures, and now it pretty much looks like a clover leaf here.
Right.
And eventually all these parts are going to separate, and they will start to float and do what they have to do. They will again start their own pleomorphic cycle. So this, I think, is a pretty good example for the people to understand what are we suggesting by the pleomorphic changes.
Oh, that was a very good explanation. Mrs. Levesque, once Mr. Naessens saw the cycle of the somatid under microscopic observation, what steps did he take to pursue his research?
Well, to tell you truly, at this point, Mr. Naessens had new information. Of course, it was very obvious that this particle was alive. It had the pleomorphic capacity and also had very numerous properties being in a resistant form. But above all this, there's something very important that struck Mr. Naessens when he looked at what was happening actually in culture. In culture, he recognized certain forms that he had previously seen in the blood of sick people. So this really, to his grand surprise, struck him very strongly. And at this point, he decided that his research, in order to clear this up, he wanted to be sure that was there any link between the presence of this particle and the state of health or the state of disease. Mm-hmm. So at this point, he figured that he had to go into a clinical trial, some type of experimental work in the field, and for this, he needed help of other doctors because he couldn't do it alone. So at this point, he really started to ask collaboration with many doctors. At that time, it was in France, of course. And he received a very good collaboration with many doctors in private labs. So let us explain here what was the idea, the research now to go on. Mr. Naessens had seen forms in live blood that he had seen, look at live blood of sick people. Now, in order to determine if there was a link between the state of health of someone and the presence of certain forms in the live blood, he needed to put in correlation, okay? That is to do side by side the same test on a big population, on many people, sick people and normal people, healthy people. And just to double check, for comparison, on a large number of people, just to see if the blood test done by conventional methods were the same, gave the same output or the same results at what he was seeing. And this was gathered, data was collected for many, many, many years, and this actually brought in a lot of information.
So while respecting the scientific approach, he, I understand, gathered a whole lot of data. Now, what did this data reveal?
Many things. First, I can maybe add that you've said a scientific approach, and you're right, because in order to, to follow the real standards of scientific work at this point, This study was done in double-blind, what we call a double-blind study. That is, the patient that was sending in his blood was sent blank. Mr. Naessens didn't know at all the person. He didn't know what was the health situation of the patient, and neither did the patient knew that his blood test was being compared. So actually there was no interference. There was no bias possible for Mr. Naessens to see what disease or not did the person had. Now, of course, once the blood test was done, and that's where the need of other doctors was so badly needed, once his blood test was done, then he could return to the medical report of that patient and see what was the final diagnosis, that conventional blood test. And that is where finally that good information was revealed. And maybe in order to talk about this, we could probably go back on the screen, take a look again at the diagram we had the previous program when we spoke about the Somatidian culture, and we could probably work from this diagram. So we might just have a little slide here on the screen.
Okay.
Now, what we can say, as we have said earlier in the cycle, when the somatid starts, it's very simple. Now, what was seen, okay, very clearly by the data of the thousands of pupils and thousands, more than 15,000 specimens were analyzed then, is that when someone is in good health, the polymorphic forms of the somatids are arriving in a certain fashion. When one person is sick or is just about getting weaker, not necessarily sick yet, but just in the midst of having a weaker natural defenses, a weaker immune system and all this, we can see a new pattern of different figures happening. So if we looked at the diagram, it's probably easier to understand. Someone in good health most likely would have in its lifeblood the somatid, the spore, and the double spore. If someone is either sick or in the process of going into disease of whatever kind, whatever type of degenerative disease, you might see a combination of 1, 2, or 3, or many forms, starting from the bacterial form up to the mycelium form. So what really happens is that in the blood, Mr. Naessens, with the collection of all these tests, have found out that between the phase 3 and phase 4 of the cycle, there is what he has called a gate control, a protection gate that really stops, halters the polymorphism of the somatid. So of course we say gate, it's a concept, it's just something for us to better understand. But bottom line, all this gate is a chemical. It's a chemical organization of the body that keeps the things, all the metabolic activities arriving in a certain way that harmony exists into the body. So what we call the protection gate is a combination of hormones and enzymes and many, I would say, chemical reactions occurring in the body, but stopping the polymorphic activity of the somatid. Now, if someone is getting weaker and weaker natural defenses, not even with a confirmed diagnosis yet, but at the time where he feels he's tired, he feels there's something wrong, something is not going well, but not having yet a confirmed diagnosis, we can still see at that stage new forms, polymorphic forms, starting from the phase bacterial form up to the mycelium form.
Do these forms identify, let's say, the degree or the advancement of the disease, or that, as these forms get larger and larger, you are sicker, if I can say this?
Well, in a way, yes and no. I mean, this particle changes form, and of course, the more advanced you are in the process of disease, most likely you will see the advanced form. But I think this question is important because, as we've said earlier, the name, the name of different figures that were given by Mr. Naessens were given according to the form, the shape, the structures of the shapes of the different forms occurring. And even though it is called a bacterial-like form, it has nothing to do with a bacteria. It's not an outside bacteria.
Just a way for him to identify
It was
that?
a way for him to identify it and also to be able to speak with scientific people, because these terms are already known. I mean, a bacteria is something very well known, yeast-like form and mycelium form, so it was just a way of putting names that would make the dialogue easier with the scientific community, actually. One thing also important that was found in blood, a little bit different from the culture, is that the resistant form, you see, that's being created from the ascus form, is not, we don't see this in the blood. There's no resistant form, really, that occurs in the blood. So all we can say, well, something very important from that experiment is that there is a resistant form, the only resistant form that exists really in the blood is the somatide. All the other forms are very vulnerable, and if you change the environment, they disappear, all these, the advanced form. So if you're looking at the blood of someone healthy, most likely you can expect to see the somatide.
And this is
the
always
spore.
alive.
This is always alive, and this is what we find in normal blood, and this is what is needed for proper cellular division. This is what is needed to sustain life, life being the growth and the repair of our different cells. So again, important to understand and to just to keep in mind that in the blood there is no resistant form except the somatide. The somatide is the only form. That doesn't change. The others can always change or disappear depending on the chemistry of the blood.
Okay. Now, you speak about a protection gate. Several times you've mentioned the protection gate. Could you explain to us again a little bit more what you mean by the protection gate?
Yes, I guess the protection gate is a subject that we could talk and talk for probably months, because this is the process actually that makes a difference, that it's the line, it's the borderline between a state of health and the oncoming stage of disease. So this is a very fine line where the chemistry of our body is either in harmony or is slowly getting out of balance. Now, with many clinical experimentations, Mr. Naessens has found out with time after, because this was done on a long period of time after the knowledge of the somatide, of course, that there's several factors that can very easily break this protection gate. And I guess this is very important to understand and to know, because it is things that we can do and things that we can change to prevent. So maybe just very briefly, because I know we could speak about this for very, very long, but just for the benefit of the...
You're going to tell us a
yes,
few?
yeah, well, exactly.
That
I
would
think
be
it's
very interesting.
It's so basic. First of all, certain things that are very important to the gate control, of course, is the physical environment. Everything that we could find in the environment that could be toxic, okay, toxic product, toxic according to one's chemistry, because what's usually toxic for one, unless it's a very toxic product, but something that is not compatible with one's own chemistry. Now, this could include food, it could be allergies to food, it could be...
Even cleansing products
It
at home?
could be any type of cleansing product, strong product, anything that we could consider toxic. It could even be an overdose of medication if this medication is not properly integrated by your own chemistry. So this is a very important point. A second point that's very important to keep this gate strong is the emotional field. I mean, if you're having different emotions, like the emotions of, I would say, death and all the new emotions that really brings you into a shock. If you lose a job, I mean, if, you know, something that's... anything that brings, a sense of loss is very, very strong and could easily... very easily.
Really?
Very, very easily. Now, of course, another thing that really shakes up the gate is if you have physical, a big shock. Let's say you have a car accident, or you have a wound, or you have a fracture, or something that would bring a big stress on the structure of your body. This also could put you out of balance. To a certain point, even surgery, having surgery, which is a stress to the body, could easily undo your gait and put you out of control. Now, other factors that could influence, we've seen also that the sensation of helplessness. If you are in a life situation where you feel that you're out of control, no escape, nothing you can do, this is very hard on your gait control and can bring you easily into the dangerous zone, leading you into disease. And finally, but not as less important, I think we have to mention that the thoughts, the way we think, the attitude we have in life, all our inner, how we feel, you know, inside, is also
pessimism.
something. Yeah,
pessimism.
This is very hard on the gait control. We've always said, we say very often that the way we think has an influence on our immune system, and in fact, with the orthobiology, we can very well measure this. That if someone has pain or, you know, any type of attitude that we have, can easily bring a breakdown. So all this, it's a very brief resume here of the different things, but I think it's worth mentioning it because if someone is in the process of healing, it's important to understand that we don't catch disease, we create unconsciously or consciously a state of disease that progressively brings us into a difficult situation.
Well, we've talked about the negatives, you know, the things that can break down that gait. And contrary to that, I would think that if someone has a very positive attitude on life, can overcome a lot of situations and protect that gait, would you say?
Yeah, that's right. I think we need to be careful here when we talk about the gait. We're not saying that all emotions that we have are putting necessarily out of balance.
It could.
But it could. But I would, you know, I think that the positive attitude is very good, and probably good emotions as love or to be happy or, you know, love in general, not only, but love of life, love of work. The whole attitude of the person is something that is highly beneficial for the gait control and to be in good condition.
Well, thank you. That was just great explanation. It enlightened this protection gait. Now, I'd like to go on to the next issue or topic. What are the major applications of this discovery?
Well, the Somatidian Orthobiology, the discovery of the somatid and the knowledge of all this particle could certainly bring a lot of applications in different fields. I mean, it could be used in various scientific fields. But because Mr. Naessens was highly interested into the blood, you know, hematology was his first interest, well, he developed all his applications in public health. One major applications, as we have just discussed, is probably his preventive screening test, which is still an experimental test, not yet available to the public, but he's working very hard to have it easily accessible at this point. And I would say that the second application of this knowledge, Mr. Naessens has designed and manufactured a product that has this property, this capacity to bring up this gait control in a very short time and permit the body to be back into the healthy range. Now, this product has been on the market since 1990 in Canada here under the Emergency Drug Service, and it is called 714X. Now, I think this is the two major applications that Mr. Naessens has been doing with his research, but of course he is still actively doing research. At this point, we're highly working on the chemical nature of the particle, which has not been done before. And there's good leads at this point indicating that the somatid is a carrier of genetic information, although it does not contain DNA. So we're working very hard right now on some new hypotheses and a new explanation that probably somatid is probably a precursor to DNA in the whole evolution of life. So this is pretty much interesting.
You're talking about worldwide protection now with the special tests. But how are we going to arrive at worldwide prevention? Is Mr. Naessens the only one with this special instrument?
I think the question is very good. No, of course, we've talked a lot here about the somatoscope, but the somatoscope is the one and only tool that Mr. Naessens is using to correct the situation he actually put into market since 1991, actually. what we call a dark field condenser, an ultra microscopy dark field condenser, which permit anyone who has interest in the work of Mr. Naessens to just take this condenser, hook it on a normal optic microscope, and anyone can easily look at the cycle of the somatid and continue research. So I think it will be more and more available for anyone who has
This
interest.
is absolutely great. This is wonderful. Thank you so much. It's been a real pleasure of having you with us on these 3 programs, and we look forward to seeing you again. Mrs. Levesque, during recent conversations, you mentioned that Mr. Naessens has designed and manufactured a product that has an impact on the gait control. Would you please speak to us about this product?
It's right. In the last discussion we had, we've spoke about the Somatidian cycle, and we explained, I think, very clearly the gait control. Now, the product that we are talking about at this point is called the 714X, which is a health product designed to replace, to build a new gait control in the event that the one that we usually have, the chemical compounds, are exhausted. Now, this product is not the only product Mr. Naessens has manufactured, but it's the one that it's available now. This one was marketed since 1980 actually, and before there were other products called GN24 and anan, which were other products that were designed to help in the same situation. Now in this case, this product now that we have is a product that has the unique property not to destroy the immune system, and I think this is very important in the intention of Mr. Naessens producing health products. He didn't want to bring in something that would put out of balance the gait that's already weakened. So we can say that all health products introduced by Mr. Naessens are designed to help the natural defenses to start to pick up, and including, of course, the immune system.
And what does this product do?
Actually, we can say that there's 2 claims, that there's actually 2 functions that this product creates once it is introduced in the body. It stimulates a deep cellular cleansing. It really starts a deep cleansing process. The cells start to clean themselves, and also it permits a cellular repair because it brings in new elements, like it's what the cells need to repair. In the circumstances where there's cells that were damaged either by disease or whatever, treatments or... So actually it cleans and it helps to repair. That's the 2 major claims the 714X has.
And what does this 714X contain?
714X is made with natural ingredients. Its prime ingredient is camphor and nitrogen. This is the prime ingredient. Actually, camphor is not the active ingredient. It is like the taxi. It's a carrier for the nitrogen element to be brought easily into the circulation. There's also some salts, mineral salts, that are 2 types, ammonium chloride and sodium chloride, which are also part of the ingredients in the product.
Mm-hmm.
And in smaller quantity there is also trace elements, which are mineral elements that are necessary to help the cells to communicate their electrolytes, that are very useful to bring harmonious communication between the cells. Mm-hmm. So basically it's all natural elements and ingredients that are brought into the circulation to help the body to clean itself. So we cannot say that the 714X is aiming directly cells that are to be taken off or bad cells. You know, it is a product that upholds and really enhances the natural defenses.
And how does it work? What does it do actually?
Maybe for the understanding of how it works, it's interesting and maybe useful to know how the product is introduced into the body. Actually, it is the basic treatment of 714X is given through injections,
Mm-hmm.
perinodular injections that are given directly into the lymphatic system
And what
Okay.
is this system?
And let us,
a few more details.
yeah, maybe we can just remind that the lymphatic system is a parallel system going side by side with the bloodstream, and actually it's the place where blood cells can evacuate all metabolic waste products that are being created by different activities in the body. So the lymphatic system is a little bit like I would say the vacuum cleaner in our body. It really takes what's not needed, the extra and all the waste product, and carries this into the venous circulation for it to be carried away by the elimination organs. So actually, its mode of intromission is very unique because it directly is injected into the lymphatic system, and this helps the body to clean itself. Actually, it is because it is given in the lymphatic system that it starts a completely new cleansing process into the body.
We know how it works through your explanation. Now, how do you administer the product?
Actually, the basic treatment, because we need to get into the lymphatic circulation. So in order to do this, of course, we could probably see a diagram to illustrate this process. But the lymphatic circulation is divided in two. There's a small lymphatic circulation which covers the upper right side of the body, and there's the large lymphatic circulation which covers the bottom part and the left side of the the body. In order to reach the large circulation, we use the perinodular injections. The product is injected daily on a 21-day basis. This is what we consider a cycle. So it is really by injection. Now, if it's needed, depending on circumstances, if we need to go and reach the little lymphatic circulation, in this case, we have to use an ultrasonic nebulizer. That means the product is inhaled and absorbed through the lymphatic system, but through the inhalation done through the respiratory tract. So there's actually two modes of introduction of the product, but the prime basic treatment is always the perinodular injections.
And if I'm not mistaken, I think you have a video that shows how to inject the product, right?
Yes, you're right. In order to do this technique properly, we have an educational tool that can be used for people who want to use it and explain step by step the different situation. Also, I mean, if you're right-handed, how do you handle the technique? If you're left-handed, how you handle, because it's you know, details and technical procedures that we want people to do properly and to be comfortable with this technique.
One question that might be interesting is, are there any side effects?
It's a very good question, because as soon as we talk about a health product, of course, if there's good signs, is there any side effect? Right? The 714X was purposely created not to have side effects. So it's not containing any chemical products that could be harmful. So it's a very simple product from that point of view. There's no specific side effects, but the only awareness we're asking patients to have is to make sure that they're doing the injection properly. That's the only thing we are concerned, because the product as such, if you do it purposely, it's really a non-toxic product.
So there's a proper way of doing it. And
there is, and we need to respect all the steps, and if you follow the steps as it is proposed in the protocol, I mean, the injection is something easy.
All right. Now, what are the effects of the 714X on the protection gate?
Okay. This is quite important because, as we've said earlier, the product was designed purposely to do this specific action. In this, you understand that the product Mr. Naessens designed works hand in hand with the preventive screening test also. So the two applications he made from the Somatidian Orthobiology are something that walks hand in hand. Now, if you do a live blood test and you see that you are out of your gate control, I mean, you're sliding slowly into what we call the dangerous zone. Okay, your gate control is down. It means that your natural defenses are getting weaker and weaker. Now, this is probably the best time for one person to take the 714X, because this would re-put the gate back in a good, strong position, and it is not given too late because there's actually no localization of disease yet.
Right.
So actually the product was purposely designed to be given in that period so it could be used on a preventive fashion. That's the whole idea. But, you know, in life, all is not white and all is not black.
Right.
We have to live with different circumstances.
Gray areas.
Gray areas, you're right. And if in any event someone is already has a confirmed diagnosis of a degenerative disease, 714X can also help. But you have to understand that the further down the road you are in the disease state, the more time it will take for your body to clean and repair, because there's more and more damage.
Right.
So the length of time you'll be using the product and the time that you will see that you will be back into a good position, well, this will be need to be considered also. So I think this is pretty much the action it has on the gait control, and from that point of view, it becomes a very interesting preventive product.
Thank you, Mrs. Levesque.